survive, we have to change behavior, and behavior has been really created by the culture and the civilization. The way out is to break the system and build a new one. Yes. And we're in that process, so I appreciate that very much. Well, I'm going to try to do a little bit of my 12-hour lecture in 15 minutes. And I'll hit some points, and if you can connect the dots, there's an amazing story here. So the first thing I'd like to talk about as a cellular molecular biologist is the nature of cells. When we look at cells, we recognize there's a physical structure here. The structure of cells are due to proteins. There are over 100,000 different protein molecules, and they're like a giant Lego set. You assemble the proteins and you create a muscle cell with some, a nerve cell with some other ones, a skin cell with some other proteins, etc. So basically, the most important insight is this. Anatomy comes from the structure of proteins. That's what your body is all about. Now that you understand that, we're really ready for something like this, the secret of life. That's all it took was one slide. Now we're ready for the secret of life. And I said, what is the secret of life? Well, the interesting part is we can look at these protein molecules. These are 3 out of 100,000. I say, what's relevant about the proteins? I say, they can complement environmental signals. There are signals that will be resonant with the proteins. And when a signal binds to a protein, it causes the protein to change shape. The shape change of a protein is where life comes from. It is the proteins changing shape that provides for all of our cellular functions. And I say, why is this relevant? Because then it gives us 2 insights into the nature of life. First, there's an anatomy made out of proteins, but then we also recognize this. Physiology comes from the proteins. And to understand how this works, there's a very simple equation. And that simple equation is this. Signal plus protein equals behavior. That if you're a protein body and you don't have a signal, that's called a cadaver. There's no behavior, but there is anatomy, okay? So where does the behavior come from? It's the binding of an energy signal. And I have energy in actually chemistry, but as we've talked about in here already, the physical world, the material world is an illusion. It's just another form of energy. So it's an energy signal that is picked up by proteins, causes them to change shape, and the shape change creates behavior. But to recognize this, the proteins are inside the cell, and the signals come from the environment. So somewhere along the line, the signal has to transport itself across the membrane to affect the proteins to create life. Well, when I first started understanding the nature of the environmental signals, that was 52 years ago. I was cloning stem cells. At the time, I was teaching medical students and people about genetics and controlling life. And what I really showed in my own research was simply this. Genetically identical cells placed into different environments express different fates. And why that was so critical was, well, the control didn't come from the genes. It was coming from the environment. Well, my colleagues, or everybody was into genes at the time. I was sort of standing on the side as all my colleagues were like genetic lemmings. They were all going toward the cliff. And I was standing on the side going, I don't think it's the genes. And they, of course, outvoted me 1,000,000 to 1. So I had to come up with an understanding of how could an environmental signal control all this. Well, basically, it led me to understand the nature of the cell membrane. The cell membrane is basically the same cell structure for every living organism. It's a bimolecular lipid layer. It's a crystal. And in the center of this phospholipid molecules, the yellow part, that's where the lipids are. So the membrane creates a barrier, but it's a non-conductor because the lipids won't let things in water go through the membrane. So a crystal membrane, non-conductor. However, what we couldn't see in the electron microscope turns out there are molecules built into that membrane, proteins. And there are 2 major classes of proteins that are built in. One class is called the receptor, which of course means what? Receiver of information. And the other class of proteins is called an effector, in this case a channel, proteins that make an effect. And when you put the 2 together, what you actually end up with is something as an information processor. The receptor receives an environmental signal. The channel translates that signal into a response that goes into the cell, and then that's what activates the proteins the proteins create behavior, and the proteins regulate genetics. So genetics are regulated by the proteins, which in turn are regulated by the environment, and so all of a sudden that's the new science called epigenetics, control above the genes. Well, the information processor changes my definition of the membrane a little bit. Before it was just a crystal non-conductor, but now I've got receptors in there. There's a synonym for the word receptor, gate, a gate. And I said, and the channel. I said, well, the channel is a protein that opens up and makes a pore, and things can go through. I said, can everything go through? No. Sodium channels let sodium in, calcium channels let calcium. So anything that can get in has to go through a channel. But the point about it is, it doesn't let everything in. Well, the membrane is no longer a non-conductor, and it's not a full conductor. It's semi-conductor. And I said, what the hell was that all about? And I said, in 1985, I was sitting there trying to put the picture together to show my colleagues how the signals did this, and I wrote this definition, and my entire world went upside down, because here's the definition of the cell membrane. The cell membrane is a crystal semiconductor with gates and channels. And why that blew my mind was because in 1985, at that minute, I said, oh my God, I read something like that just recently. Where did I read that? And what I started to recognize at this point was I just bought my first Macintosh computer. I had a book from Radio Shack, and the book from Radio Shack, and the introduction of understanding your microprocessor, a chip, definition, a crystal semiconductor with gates and channels. I said, wow, what a coincidence. But then I'm going to tell you the truth because we're running out of time. It is an exact homologue of a computer chip. It is exactly every part in a silicon chip and every part in this protein chip are one-to-one exactly the same. And the point about it is this. The membrane of the cell is an information processor. The nucleus of the cell is a hard disk with programs. We used to think it was read-only. Your genes are your life. But now we recognize it's read-write. You can change the genes. You can make over 3,000 different proteins from the same blueprint, the same gene. And all of a sudden it says, and what's regulating life is not the genes, it's environmental signals that do that. So the receptor is a stimulus, the channel is a response, but in computer technology, the receptor is an input, the channel is an output, I/O, bit of data. That is a bit of data. I say, well, what is it? I say, well, the name of it is a receptor, effector, integral, membrane, protein, complex, and everybody goes, ooh, big deal. So that's not what I'm going to show you. Let me first show you how it works. Very simply this. The receptor scans the environment for a signal. The signal shows up, causes the receptor to change shape, allows it to couple to the channel, which changes shape, and the channel sends a signal inside the cell. That signal controls genetics and behavior. When the environmental signal's done, the switch is broken, the signal shows up, the switch is connected, and the behavior or genetics are then expressed by this right now. So I say, so what's the name? I say, well, I give you that stupid name. It doesn't mean anything to anybody. But let me give you a functional name. This is a switch. 100,000 of them on the cell membrane at any one time. There's receptors and channels. I say, what's the function of the receptor? Interesting point. Where are your receptors? Eyes, ears, nose, taste. They're all in the skin. You and the cell are fractal images. Different story. Come back later. Okay. And I said, well, let's define it by its function. I said, what's the function of the receptor? And the receptor is awareness of the elements of the environment. Awareness of the environment. Eyes, ears, nose, blah, blah. I said, well, what's the function of the channel? Oh, that sends a physical sensation into the cell that controls the behavior and the genetics. So I said, wow, the definition, wake up because it's a cool moment. The definition of the switch, let me emphasize, the switch is the interface between an environmental signal and a cellular behavior. And I said the switch, definition, awareness of the elements of environment through physical sensation, and then we go to the dictionary and darn it, look at that word, perception. Awareness of the elements of environment through physical sensation. What you are looking at are protein perception units. And perception units are the foundation of consciousness. Consciousness is collective perception. So how does a cell work? And the answer is the cell works because environmental signals from the outside adjust the function of the inside through the bits of data built into the cell membrane. Now, if you understand this, it profoundly changes our understanding of evolution. Evolution is not based on genes. As we already heard, the human biology has as many genes as a worm called Caenorhabditis elegans, which is 1,271 cells. They have about 20,000 genes. We have 50 trillion cells, 20,000 genes. Genes RNA. What it is the chip. How many bits of data can you put in a chip? And that is the element of an actual mathematical equation of consciousness. I say, where's the math come from? The size of the proteins and the thickness of the membrane are predetermined. You cannot stack perception units up in a membrane. You can only lay them flat in a monolayer. And you go, what the hell does that mean? I go, well, then the number of perception units is directly related to the surface area of the membrane. And all of a sudden now you have a metric to measure consciousness, membrane surface area. And there's a repetitive pattern of evolution, starting with bacteria, which have limited membrane because of an external capsule, breaking into an amoeba, which has an internal skeleton making a big membrane, etc. And then into the fact that we are made out of, what, 50 trillion programmable chips called cells. And then I say, where's the programming come from? Well, I already showed you, the perception switches do that. Yeah, but here's the most important part that I want to go with right now, and that's simply this. There are no two people that are biologically identical. I can tell you that just because you try to take your cells and organs and put in somebody else, their immune system will reject, not self. All of a sudden it says, oh my God, then if I put cells in your body, your immune system can tell me that it's not your cells, but they're my cells, then damn it, there must be some identity as to where the heck the identity is. Because the cell immune system can read the identity of the individual, recognizing there are two people with the same identity. Where is that identity? Well, it's based on switches, perception switches. Just a little thing about perception. Perceptions control biology, but not all of our perceptions are accurate. So a better word is biology is controlled by belief. That's the belief that you send to the receptor. Right or wrong, it's going to make a response. Now let's go back to the cells. Why am I picking 2 liver cells? I just want 2 identical cells. 2 brain, 2 muscle, 2 skin. I don't care. I want 2 identical cells because functionally and cytologically they're almost exactly the same. But what's different here? Well, one's Bruce's cell and one's Margaret's cell. I can't put my cell into Margaret's body. Her immune system rejected. Margaret can't put hers into mine. Immune system will reject it. The immune system knows where the hell the self is. I say, so where is that cell? So here's my little ploy. I'm going to remove the color from the slide where the cells are essentially almost exactly the same, whether Bruce or Margaret's point, leaving the color behind where 2 people differ from each other. And boom. Where's the difference? The outer surface of the cell membrane. Guess what? There's a group of these perception units that are called, in medicine, a small number of them, self-receptors. Self-receptors, receivers of self. Guess what? No 2 people have the same set of self-receptors. And I go, so what the hell does that mean? Your identity is marked on your cells. The exclusion, red blood cells. That's why you can take blood from other people. There's no identity to it, okay, except APO. But the reality is, each one of us has a unique identity. You know where that identity is? On the surface of your cells, and it's a group of large number of receptors called self-receptors for a portion of them. I say, why is it relevant? Because where are the receptors? Outside surface. So where the heck do you think the idea of self or the signal of self is coming from? Not inside. It's being picked up by the antennas on the outside. So the relevance is this. We, identity, physically, self-receptors. But source, environmental signal. We're not even in here. We are picked up by these receptors. Your identity is out here, and you're playing through this. Now, in the first, do I have any minutes left? I have more than one time. Oh. Because as a scientist, I never believed in spirituality. I didn't have any role of consciousness. I was being dragged into this field by my cells. one instant of time, I was not spiritual, and then 1 second later, it was like, of course spirituality is not even a question. I'm not in here. I am the broadcast. Well, then all of a sudden my conscious mind got engaged and said, then why have a body? Why not just be spirit? And the answer came up from 50 trillion comedian cells. Obviously, there's some Jewish genes in there, because I asked a question and the cells responded with a question. I said, why have a body and a spirit? Why not just be a spirit? And here was the response. Bruce, if you're just a spirit, what does chocolate taste like? And that was it. I said, oh my God, the biology. The biology is a spectrum analyzer. All the receptors are reading different parts of our world and turning it into something that imagination couldn't see. But it's a mechanism. What does love feel like? What does chocolate taste like? These are biological responses that are picked up by the nervous system, translated by the nervous system, and here's the cool part, sent back to source. Sent back to source? Yes. How do I say that? Because if I put wires on your head, EEG, electroencephalograph, I can read your brain activity. And that was the belief that says, oh, the activity is encased inside and controls everything from the head down. But there's a new device. It's called magnetoencephalograph. I say, why is it relevant? The probe doesn't even touch your body. The probe is out here. I say, what does that mean? My God, my thoughts aren't contained in my head. They're broadcast back out in the field. That's what magnetoencephalograph reveals. Your thoughts are not contained in your head. Your thoughts are a field. You're generating fields with your thoughts, which then come back and may haunt you. So the bottom line here is, how does all this work? So I said we've got the proteins built into the membrane, fine. Then we have the signals that come from the environment. When the signal binds to the protein, and it's like a lock and key, different signals, different proteins, when they bind, they create behavior. That's where life comes from. Life is protein plus signal. Now the question is, well, what the heck kind of signals could we be using? And here comes a wonderful story. And it was a wonderful research from a couple called Popristic and Goodman, published in the journal Nature. They were working on the twisting of an ethane molecule. An ethane molecule can twist and change shape. They go, why is it relevant? It's the same mechanism that proteins use to change shape. So if you can understand how the ethane molecule is rotating, then you have insight into how proteins rotate, which is where life comes from. So here's the point. Popristic and Goodman are looking at the staggered structure of ethane, meaning it rotates under different situations. Point. The idea of science is predictability. If you can predict the movement, then you have insight into how it works. So what did they do? Popristic and Goodman used Newtonian classic physics principles, energy and localized fields, to measure what is causing the change of the ethane. The idea is if you understand how it works, you can tell all the intermediate structures from shape A to shape B, you can predict the movement. Well, it didn't work. They couldn't predict a damn thing about it. What did they do? They had to go into the principles of quantum physics. It turns out that the rotation of the molecules that generate life are driven by quantum physics principles, not Newtonian physics at all. And I say, well, why is it relevant? Well, there was an editorial in the same journal from Frank Weinhold, a chemical physicist, I love it, A New Twist on Molecular Shape. And I said, well, what is the point? I said, well, just the subtitle. What are the forces that control the twisting and folding of molecules into complex shape? Why even ask the question? Answer is relevant. If you understand the forces that cause the twisting of molecules, then you understand the forces that cause life. That's basically what it was. And then what does Frank Weinhold go on to say? He says, don't look for the answers in your organic chemistry textbook. And I say, what's the relevance of that? And I say, organic chemistry is the foundation of allopathic medicine. Yeah. And it says you want to understand how life works? Do not look in that chemistry. So basically it says the concept of materialism, which is the drug company's whole strength, is it's out of here. And I go, so relevance about that? I say, well, Back years ago, there was an article on the difference between a signal that's a chemical signal versus a signal that's an energetic or resonant signal. And this is from a Clare biophysicist, University of London. And what did he say? He compared the effectiveness of sending a signal via energy or sending a signal via chemical. Here's the point. When chemicals join to another chemical, they give off heat. It's called the heat of reaction, thermochemical reaction. I said, what's the definition of heat? Dissipated, wasted energy. So when a signal as a drug binds to a target, it turns out 98% to 99% of the available energy of the molecule is dissipated as heat. So a chemical signal carries less than 2%, around 1% information. The rest of the information, the energy is lost. When he compared it to resonant vibration, 100% of the signal was picked up and received with no loss. So basically it says, in competition, resonant molecular machines would undoubtedly be selected over conventional thermodynamic machines, which is another word for pharmaceutical drugs. Johns Hopkins University, Richard Conn Henry, The Mental Universe, just the first sentence in the subtitle. The only reality is mind and observations. Let me give you the last sentence, and my conclusion is his conclusion. And this is the actual printout of the last sentence in this article in Nature. The universe is immaterial, it's mental and spiritual,